Good afternoon, everybody, and welcome to this Alumni Festival event, Unlocking the Hidden Complexities of Evolution. My name is Michelle Cooper, and I'm privileged today to host this event on behalf of the university as a member of the Alumni Advisory Board. So a little bit of housekeeping before we get started. We will have a session of Q&A at the end, so we will have roaming microphones. If you have an opportunity to ask a question, please wait until the microphone comes to you so we can all hear you clearly. This event is also being recorded and it will be available after the festival for you to indulge in again and share with others. So we'll do our best to address as many audience questions as we can at the end, but we do have a limited time as well. So, the University of Cambridge has been the cornerstone in the study of evolution. From Darwin's early observations to today's sophisticated genetic analyses, the research that takes place here at Cambridge University continues to push the boundaries of our understanding of life's complexity, the mechanisms that drive evolutionary change, and the challenges and opportunities ahead of us. Today we will hear from three leading academics: Dr. Emily Mitchell, Professor Ian Henderson, and Dr. Aylwyn Scully. Each of our speakers will have 10 minutes to share some of their research as they illuminate the past and help us consider how advancements in their respective fields will inform the future. Following this, we will, as I mentioned, have an opportunity for a short Q&A session and also a small panel session. So, ladies and gentlemen, it is my pleasure to introduce our first speaker, Dr. Emily Mitchell. Dr. Mitchell is a pioneering paleontologist, paleobiologist, and ecologist, and her research spans deep evolutionary time, from the first animal communities to modern ecosystems. Using innovative laser scanning techniques and interdisciplinary methods, she has broken new ground in understanding early animal life and their environments, extending her studies to deep sea and Antarctic organisms. Please join me in welcoming Dr. Mitchell to the stage.
Hi, it's really lovely to be here today with Ian and Aylwyn to talk to you about what we do as biologists to try and unlock the hidden complexities of evolution. So I'm a paleontologist. I work in the Department of Zoology and the University Museum of Zoology here in Cambridge. And my research is mostly interested in understanding the origins and early evolution of animals. Now, this is a topic that fascinates many people over a very, very long time, including Darwin. And Darwin actually mentioned it in The Origin of the Species, and it was a cause of concern for him when proposing his ideas of natural selection, because, as he spoke, there is another and allied difficulty which is much graver. I allude to the manner in which numbers of species of the same group suddenly appear in the lowest known fossiliferous rocks. So what did he mean when he said this? He was talking about what's known as the Cambrian explosion. So this is a period of time, about 520 million years ago, where all across the world you suddenly see huge amounts of fossils appear in the rock record. Things like trilobites and trace fossils, as well as worms and swimming arthropods and sponges and corals. Now these were troubling to Darwin because, as it appeared at the time, there was nothing in the rocks before them. They just appeared out of nowhere. And if his ideas of natural selection were correct, in that it means you should get slow adaptation to the surroundings, to the environment, and evolution should happen gradually over time, large complex things should not suddenly appear out of nowhere. Now, this Darwin's dilemma remained unsolved for almost 100 years until the 1950s. And it was here in the UK, in Charnwood Forest in Leicestershire, that two school children found the first fossil in known Precambrian rocks. They were Tina Negus and Roger Mason. And they found this wonderful fossil here. You can see left-hand side. And this was really, really exciting, because what it means is that instead of having suddenly these Cambrian fossils come out of nowhere, you suddenly have your fuse to the Cambrian explosion right in what's now known as the Ediacaran time period. But what these were was really difficult to work out. So you can see with the Chinia, it's got branches that have branches that have branches. And these are known as fractal-style branching. And this sort of fractal branching you only find in the Ediacaran. You don't see it in any other body plans elsewhere in the fossil record or alive today. Now, while it looks superficially like a plant, the rocks it was found in can only be found in deep water. So we know it wasn't. But it's not just this Charnia that's difficult to understand. We can see a variety of other different Ediacaran fossils here. We've got Dickinsonia in orange. So this is a large flat organism, but it can reach over 1 meter in length and almost as wide. We also see a huge amount of different radial symmetries. So today we have bilateral symmetry, that's us, and we have fivefold symmetry, so that's things like starfish. But in the Ediacaran, we had 3, 4, 5 as well, but 9, 11, 13, lots of different types of symmetries. And it's these sorts of things that are quite fundamental to what an organism is that makes them really hard to understand. And indeed, they're so hard to understand with so few analogues to modern organisms that for a long time people thought that the Ediacaran organisms were a failed experiment in evolution. The idea that as life got large and complex, but ultimately failed, everything went extinct and gave rise to the Cambrian, and the animals be able to evolve then. But we know now that this isn't really what's going on. So in the last decade or so, there's been a huge suite of different modern technologies that have enabled us to understand Ediacaran organisms in a very different way. So there are things developed by Imran Rahman in Natural History Museum using computational fluid dynamics. So we can see here one of his simulations. You simulate how the fluid flows, the water that these animals lived in, flowed over them and around them. And this helps us to understand how they're interacting with their environment and how they're feeding and how they're interacting with each other. So my work utilizes laser scans. And so we sit on rock surfaces in Charnwood and in Canada and Newfoundland and laser scan entire surfaces in order to capture these fossils down to a micron level detail, sub-millimeter detail. We also have some really, really amazing work from colleagues in Russia, Ilya Bobrovsky, who managed to find some organic remains in some Ediacaran fossils. So almost 600 million years old, they managed to find these lipid biomarkers, these leftover fossil chemicals, and compared them to different modern organisms. And what this means now is that we can be sure that there are Ediacaran fossils that are animals. So here we've got your Dickinsonia, and what Ilya Bobrovsky did is he compared the cholesterols, the sterols of this Dickinsonia to different sorts of plants and fungi and bacteria and animals. And what they have in common is the same sort of chemicals that you find in animals, but not other creatures. And also, to kind of further advance this, he looked at other sorts of Ediacaran fossils, so simple disc fossils, and showed that they were cyanobacterial, not animals, and also managed to resolve some of the gut contents of some of the slightly younger fossils. There's also been advances in Ediacaran paleobiology by looking at how animals develop through time, how they grow. And so Frankie Dunn at the University of Oxford Natural History Museum looked at the different traits that allow things like Charnia to grow. And what she demonstrated is they didn't have any common points with things like fungi and plants, but did have some in line with sponges and with other animals such as cnidarians, which means they can be broadly placed as animals. So now we know that they're animals, how can we go about researching them, given that we know that their morphologies, their body plans are very, very different? And this is problematic because a lot of paleontology, especially historically, was done by comparing the body plans of fossil organisms with those of modern ones. But if you've only got a very limited number of points, how do you go about it? So what we do is we take a different approach. So in the Ediacaran, you have really exceptional preservation. Here we can see some fossil sites at Mistaken Point in Newfoundland, Canada. And these are UNESCO World Heritage Sites because of their importance to understanding the history of life on Earth. Now, these are soft-bodied fossils. They predate the ability to build hard parts like bones or shells, but they were preserved under underwater ash flow. So it's a bit like Pompeii in that this ash came down and killed everything where it was living. They also predate the ability to move, which means that they were literally the entire community was preserved where it was living on the sea floor. And because you have these thousands and thousands of Ediacaran organisms, it means that you can use a whole host of quantitative, ecological and evolutionary analyses to try and understand what's going on with these Ediacaran organisms in ways that you can't normally do in the fossil record. So what my work has done is, as we go throughout the Ediacaran, we start with the very, very first animals. And what we find is really quite surprising because there isn't much going on in terms of competition between different Ediacaran animals. So So these lived on the seafloor. If you think of something like a coral reef, everything competes with everything else for space. It's very, very crowded. Similarly, in the deep sea, if you look at the deep sea, it's very, very sparse. All the corals and sponges are very spaced out because they're competing with each other for food. If you have two corals that are too close together, there's not enough food for both of them, so one of them will die, leading to this spacing out. So you have a very competitive environment. That's not the case for the early animals. The early animals had plenty of food. They didn't really compete with each other. And so what this suggests is that, and they didn't really interact much with their environment. There wasn't a big environmental signature like we see today. So this suggests that the differences we've seen as the very earliest Ediacaran organisms are evolving aren't necessarily due to systematic adaptation to the local environment, but instead are potentially due to kind of stochastic differences within the population. This then changes as you move throughout the Ediacaran. You start to get movement, you start to get more adaptation to the seafloor and more competition between different species and within species. Then as you get just before the Cambrian, you get more specialization. You get different animals living in different places, both within kind of the broad communities and also around the world. And so if we go back to Darwin, what this has shown us is how over the last few decades we've managed to resolve Darwin's dilemma. Now, as one of our great alumni said about the finding of China, that this was a discovery that transformed our understanding of the mystery of mysteries, the origin of animal life. And through research that we're doing here in Cambridge and elsewhere, we're starting to understand what's actually going on with these early animals and what's driving their evolution. Thank you.
Thank you so much, Emily. Our next speaker is Professor Ian Henderson. Professor Henderson is a leading geneticist specialising in epigenetics and plant sciences. Here at the University of Cambridge, he leads research on genetic and epigenetic factors controlling recombination, and his groundbreaking work, including the generation of super recombining crops, earned him the 2013 Society for Experimental Biology President's Medal. Please give him a warm welcome to Professor Henderson.
Okay, so thank you, very kind introduction. It's wonderful to see so many people here. I was a bit intimidated actually when I came in to see how big the room was. But basically what I'll be talking about today is more to do with genomes. So, the sort of genetic basis of those types of evolutionary radiations that Emily was talking about. And to start with, I'd just like to discuss this idea called the C-value paradox. Now, the C-value is a measure of how much DNA is in a genome, so how big a genome is. And what you can see in the middle of this slide is a kind of graph showing genome sizes in a range of different organisms. Now, the human species would be somewhere here. Okay, so the C-value paradox relates to the fact that when we look at how big our genome is, and that we traditionally consider ourselves the most kind of complex organism, that we ought to have the biggest genome. But you can see there's actually lots of plants and animals that have far bigger genomes, much more DNA than the human genome. And so two examples of this. On the left, you can see a plant called Paris japonica, and also the axolotl. Both of these organisms have genomes like 100-fold bigger than the human genome. But we don't tend to think of them as being more complex than we are. So how can we understand this paradox? You might recognize this scientist. This is Barbara McClintock, and she was a corn geneticist, and she was studying these patterns of pigmentation in corn kernels, and she made a very important discovery that what was causing some of this patterning were genes that were moving around the genome, so jumping from one place to another. And these she called transposable elements. And at the time, this was just a discovery in maize, but over the years it's become clear that these transposable elements are distributed in essentially all organisms. And here you can see on the right-hand side this breakdown of the human genome, and so it's kind of showing the amounts of sequence occupied by these different classes. About half of the genome is non-repetitive genes, and the other half consists of lots of repeats, many of which are these transposable elements. So actually most of the human genome consists of these bits of DNA that are kind of thought of as selfish or viral-like, that are kind of colonizing the chromosome. And they are, often called selfish DNA, but they can also end up being useful in different ways. And so what this slide is showing is a couple of situations, one in grape and one in orange. And what you can see in each of these cases is a diagram representing a gene that's producing pigmentation. And so this gene is making anthocyanin, which is making this Cabernet grape nice and kind of deep purple. And what we know is that the Chardonnay grape, that you'll know from white wine, the reason these grapes don't make this pigment is because one of these transposable elements has jumped into the beginning of that gene and essentially turned it off, causing this absence of pigmentation. And further things can happen. In this case, a rearrangement occurs of that transposon, producing these ruby grapes. And you can see similar situations in the oranges where, again, these transposons jumping in and out of genes is changing pigmentation. And so we know that this happens in these cases, and because transposons are so frequent in lots of different organisms, it's true that in many species, including humans, this type of activity can have important effects. So you can see here, like they're jumping into genes and disrupting them. And so they are often thought of as being a bad thing, a dangerous thing. But we know also that they can end up being useful. So this is a bit of a complicated slide, but it relates to the human immune system. And so when you become sick, your body will produce antibodies represented by this kind of Y-shaped structure. And if these antibodies recognize the pathogen that you're infected with, it can help get rid of it. And the key thing about the antibody system is they have this variable region in this cone. And basically in your blood system, when you become infected, the antibody genes diversify and change that structure, allowing them to recognize lots of different pathogens. And the way this happens is something called VDJ recombination, where the gene for the antibody is rearranged. And the key point to this slide is that the thing that does the rearranging is ancestrally one of these transposable element proteins. So it starts off as a parasite, but in this case, part of that transposon has essentially been domesticated to do something useful. In this case, to create genetic diversity in the antibodies during infection. So for all of those reasons, it's very interesting to know about transposons and repeats and all of the totality of the genome sequence, not just the genes. And but for a long time, these parts of the genome have been called the dark genome or junk DNA, things like that. And partly that's because previous sequencing technologies had a very difficult time actually sequencing and assembling those parts of the chromosomes. But so you can imagine if you're sequencing a genome, it's sort of like a jigsaw puzzle, and you're seeing it in fragments, and then you're trying to piece all those fragments back together again. And so if the pieces of your jigsaw are very small, and you've got areas that are just kind of, like blue sky, let's say, it's very difficult to solve that puzzle. Whereas if you have much bigger pieces, it becomes a much more straightforward task. And the thing that's changed in recent years are things like this technology, Oxford Nanopore. So this is a very different approach to sequencing DNA, where these DNA molecules are being passed through this pore, and according to which base passes this kind of pinch point, there's a change in electrical signal across that pore. So the DNA kind of whizzes through, and then using this kind of electrical signal, you can read off the sequence of the bases. And it's amazing that this works just at all, but the beauty of it is that you also get very, very long stretches of sequence. So you're more in this kind of scenario, doing a jigsaw with very long pieces of sequence. And what this is allowing now is, for example, this Darwin Tree of Life project, they're sequencing every organism in the British Isles. And further, people are sequencing lots of individuals within species as well. So, the Biobank project you may have heard of. And so we're sort of moving into this realm of what you could call pangenomes. That's where every species we have the sequence of, and it's the totality of the genome that we can see, and also genomes from lots of individuals within species. And this is just an analysis we're doing at the moment, looking at a range of plants and animals across this kind of radiation, and looking at different aspects of how those genomes are organized, including things like how many transposons are there, which types. And it's just a very fascinating time to be doing this research, just because this flood of data is kind of emerging. And that's it. Thank you.
Thank you. Everyone's being really on time today. I don't even have to flag, let's keep moving. So our final speaker today is Dr. Aylwyn Scally. With a background in theoretical physics, Dr. Scally's computational expertise and large-scale genetic analysis have shed light on the evolution of human and great ape populations, including his groundbreaking work on the gorilla genome and human Neanderthal genetics. Dr. Scally also seeks broad understanding in the meaning of ancestry and how that impacts the way we see our world. Please make him welcome.
All right, thank you very much indeed. So I'm going to bring the story. Oh, before I do that, I think I better switch to my slide, hadn't I? Here we go. I'm going to bring things up to the present day, or closer to the present day, and also think to be much more parochial and just talk about our own evolution and origins. And genetics has a lot to say about that. And I want to try and talk about two things. One is some of the ways, or one or two of the ways in which our own sort of intuitive sense of ancestry, and our ancestry in particular, is actually at odds with the reality that we find, or the sort of structures of ancestry when you think about them, and the relationships between us. And the other, I want to perhaps more positively give you a sense of some of the exciting, really exciting things that we are discovering through the analysis of human genomes all around the world. And the context, of course, is that this is an incredibly rapidly growing field, and it has grown dramatically in a very short space of time. Just 25 years ago, we had no human genome sequences, and now we have countless millions of them, really, or many millions of them, countless numbers of them we really don't know, because it's become something which is ubiquitous now in healthcare all around the world, or certainly in developed countries. And it's also a big part of this global genealogical industry. So a lot of people here may well have had some or all of their genome sequenced in that context. And I want to start with that particular industry, and this is an advertisement from that from a few years ago. And I think it illustrates the kind of the way, the tone in which a lot of people approach it, which is as a sort of bit of fun. I mean, actually, that varies quite a lot from country to country. In some countries, this is taken a lot more seriously, but certainly here and in the United States, it's a kind of an amusing thing to do, or sort of entertainment, infotainment. And there are various reasons why this gentleman is obviously not, in fact, a Viking, but what he is getting at is this sense that we have of our ancestry being descended from quite deeply into the past. And a lot of people, I think everyone really approaches their understanding of their ancestry through this, their family tree, which is a kind of a familiar enough concept. And indeed, it is sort of the fundamental concept of ancestry. All of your DNA has to be inherited through this, within this. And you may well have explored your own family tree, and due to the nature of historical records, you can go quite far back in many ways, but it gets quite sort of pretty patchy, and often you can go quite far back in some lineages, but really others are totally obscure and lost in terms of the records of who these ancestors were. And that leads to people kind of thinking quite linearly about their ancestry. And you maybe go several generations back and find that there's somebody interesting in your family tree, a French Huguenot perhaps, or someone from another continent. And you might then say, that might then become part of your identity for some people. And that's perfectly fine. But there is, it does lead to some quite extreme examples. And this TV program, Who Do You Think You Are, was a good example of that, where in this case the actor Danny Dyer managed, or the researchers I should say, traced his lineage all the way back to Edward III, which is indeed something that you can do. Often it's the kings and queens who are the only people you can find if you go back this far. And sure enough, Danny appears in this get-up shortly thereafter. And it's kind of amusing. And obviously what this is ignoring is all of those other ancestors, or at least it's discounting the fact that your ancestry includes all of those other people whom you couldn't trace back to. And here's a sort of diagram of what your ancestry looks like. So here at the bottom maybe is you, individual A, and the red area is your genealogy growing as it goes back in time. On average, it's going to double every generation, obviously. Everybody has a mother and a father. And so inevitably it's going to grow. And the sort of the outer shape is essentially the size of the entire population, which we know in fact is smaller as you go back in time. So there's a point which, depending on where you are in the world, but here in Europe, typically around about 1,000 years ago, your ancestry, your ancestors are going to essentially fill that entire population. So everyone alive back then and prior to that, who has descendants today, is one of your genealogical ancestors. It's a sort of mathematical certainty that must be the case. And that's true not just of you, but of any other person alive today in this part of the world, should we say. So that you share all those ancestors. Your family tree is really a shared thing throughout most of history and prehistory. And that means it includes Edward III, Charlemagne, all the Vikings, and Cleopatra, you name it. And that's really the story of these relationships, that we're all really, almost certainly everybody here, or most people here, will have some descent from Edward III in particular. It's sufficiently far back, but definitely the Vikings. So what that does, it's interesting for ourselves, but it also undermines, I think, some notions of ancestry which are perhaps less positive. And that's when it comes to thinking about differences between groups, and in particular differences between countries. And actually we're quite used, I think, to thinking about present-day nations or present-day groups of people as collectively having these deep ancestral roots. And 100 years ago, that was certainly the predominant mode of thinking as well, and probably hard to see in this map, but this is a map from about, was it 1920 or something? This was how certainly people's understanding of the history of European groups was, that if you went back, the people living today in Germany were essentially primarily descended from these Teutons back in the prehistoric period, or the early medieval and earlier periods, and elsewhere you would go back to the Celts. And so on. But of course, really, all of these genealogies of all of these people are completely mixed and shared by the time you get back this far. So it does undermine those ethno-nationalistic ideas, which clearly had very dark consequences in the middle of the last century. Nevertheless, there's to some extent still this idea, this notion of these distinct groupings is still reified by our present day, the way that we approach DNA and ancestry today, particularly these ancestry companies, which still would like to present your ancestry in the form of a set of categories that have been predefined and give you sort of percentages of assignment to them, which is a simplistic representation of this really complicated structure. And one of the things that really has also contributed to undermining this view is the discovery of DNA, not just from lots of people today, but also from what we might refer to as ancient DNA, but it includes people from the relatively recent times, medieval periods even. And this is a picture, actually this is already out of date, this is a couple of years ago, showing where we've been able to, samples from which we've been able to get DNA, ancient samples. So they're colored by how old they are. The yellow ones are roughly correspond to the medieval period, and then further back, Iron Age, Bronze Age, going back to blue and purple samples which are thousands of years old, or tens of thousands in some cases. And it's primarily from Europe for the reasons of that's where there's been the most study, but there are other factors which make DNA hard to get from samples which are in warmer climates, for example. So there are various things contributing to the distribution of these. But really, it's quite fascinating, some of the things that we can get from these ancient DNA samples. So here's an example of a study that was done on a grave site, a burial site from Neolithic France, we're talking about 7,000 or 8,000 years ago. So we've got these skeletons here that they've dug up, and we're able to extract DNA from the bones, say in the teeth or in parts of the ear, and the bone in the ear, which apparently is very good, the petrous bone. This is on the left is a map of all these the sites or the graves in this burial site. And from that, we're able to reconstruct really this fascinating genealogy of the individuals in this burial site from 7,000 or 8,000 years ago. And you can see then really interesting things that archaeologists would never have been able to know otherwise. That, for example, here, the sites buried close to each other are indeed more genetically related to each other, and that there are different groups, different parts of this large extended family present. And also, interestingly, who's not present. Certainly types of people, for example, women tend not to be, are less likely to be buried here, and some branches of the family are excluded. And then there's also, up at the top here, a whole bunch of people who aren't in this family tree. And often what we find is that they have DNA which actually looks like samples elsewhere in the world, nearby regions or from further afield. And one of the things we can do is reconstruct then patterns of gene flow and migration all around the world. And initially, geneticists, when in doing that, you start to find this picture that actually, even we're familiar with people moving around the world today through airplanes and intercontinental travel, but actually that's something that's been going along maybe at a slower rate, but as far back as we look. And initially, people started drawing these kinds of pictures to showing, some of the movements of genetic ancestry that we've discovered in the last, say, 5,000 years, going back. And it's pretty much the case in every part of the world. The problem is that this was also far too simplistic, really, and in fact, it ends up looking a little bit like those pictures of, invasions and migration routes from that map I showed you previously, and is quite reductive in that sense. And a much more accurate picture. It almost defies kind of illustration on a map, to be honest, because if we were to try to draw all of the connections that we have discovered between groups at various points in time, it would just be a tangled mess. We end up with a picture something like this, which actually, rather than trying to show the individual movements, shows our reconstruction, tentative at first, because this is really kind of the very beginning of this kind of research. We're improving our picture all the time. But what this is showing is regions where we infer there had to have been a lot of, or relatively more, movement of genes between around the world, and those are the blue regions. And then in the brown, places which we think have acted essentially as a sort of barrier to gene flow, relatively less movement of ancestry, either directly through movement of people or just through contact from one generation to the next. And one of the really exciting things that's happening at the moment is us refining the resolution of this as we get more and more ancient samples and as we sample more people from around today. So that's some of the exciting things that are happening in our understanding of human ancestry at this time.
Thank you so much. We will have a microphone going around, but I might kick us off with a couple of questions. Each of you have really come from a very different subfield within evolutionary studies, so genetics, paleobiology, ecology, and so offering distinct perspectives on the complexities around evolution. So a question that's on everyone's mind at the moment is thinking about the application around AI. So I was just wondering, as AI and machine learning become more integrated into scientific research, How do you see some of those technologies contributing to your own fields and fostering the interdisciplinary collaboration? And do you think that AI and machine learning could help bridge some of those gaps between the areas of expertise that you all hold to reveal new insights about evolutionary processes? Big question. Who wants to start?
I'll kick off. So, it's early days yet, but clearly I think these kind of things are going to have a role because of, I didn't show some of the complex structures that we use in genetics to try and represent relationships between individuals, but they really are stupendously difficult to analyze by any other means. We have some very smart computational methods to do so, but at some level, all the different patterns that we're seeing, which reflect different events in the past, become quite difficult, too difficult really to infer sort of in a deterministic way. And that's where machine learning is potentially a huge application in terms of helping us to identify certain patterns that we see in the data with certain types of event in the past. So it's a growing field. I would say it's something where you want to be careful because there is always this sense if you've done something with machine learning, have you really understood what has been discovered? But to some extent, it's just like another tool potentially that we can use. I think it will be, it'll help us. I don't think it will change the fundamental thing that we're doing.
Yeah, I think for me, what's particularly useful, and we've started doing some of this already, is aiding with the data processing and collection, both for the modern and for the fossils. So in order to get our data, we're talking tens of thousands of fossils and modern deep sea organisms. Someone has to look at it and identify it. And this takes a long time. PhD students obviously love spending time in front of computers, but it is very, very time consuming. And the kind of application of machine learning methods to identifying kind of different sorts of biological organisms is really kind of coming on leaps and bounds. And so the amount of data that's available to us is changing really, really rapidly from a field which historically in paleo is very, very limited, where you start off potentially with this one or two specimens to thousands. So, and the more data you get, the more different sorts of analyses you can do, and the complexity of the patterns that you can pick out is better, and the kind of more subtle patterns. So for me, that's what's the kind of most exciting, is this using it to get a lot more data a lot more quickly.
Yeah, I think in genomics, there's kind of two main ways it's been used that I can think of. One is the situation where you've just sequenced a genome for the first time, and it be millions of A's, T's, G's, and C's in a string. And what you want to know is where are the genes, where are other things, and that's essentially called an annotation process. And that's been quite laborious and a lot of manual work. But if you think of how things like large language models work, you can treat the DNA as a kind of text or language and ask these algorithms to predict where things like genes are. And that actually works quite well. And as more data accumulates, it will get better. Mm-hmm. The other big one is what's known as AlphaFold. And so 20 years ago, 10 years ago, if you had a gene, and you knew that gene made a protein, and you wanted to know what that protein did and looked like, you'd have to find a crystallographer to make a crystal of it and shoot X-rays through it and then reconstruct the structure. AlphaFold is a kind of DeepMind approach, and it will just in silico predict from the DNA sequence what the protein that it would produce would look like and how it would fold. And again, it's pretty good. Not quite as good as crystallography, but you suddenly have all of the structures or ideas of what the structures would be like. So, and again, that's going to get stronger and better the more data gets fed into it.
Oh, thank you. So events like this one obviously help us communicate the science and the research that you do to alumni and others. So I'm also keen to get your thoughts on how you think we might be able to better communicate the complexities of evolutionary science, and particularly understanding that, this was a Cabernet, now it's a Chardonnay, and those little kind of tidbits kind of help us, I think, process the information in a way that's engaging as well. So how might we continue to do that and explain this type of science to a broader to a broader public audience to enhance our understanding and maybe also help us secure additional support for this type of research, because it's quite fundamental research, but often it's not always the fancy one that people might be throwing money at. So yeah, your thoughts on that?
Yeah, shall I go first? I mean, one of the challenges in my field, actually, which I suspect, or well, I hope the others don't have as much, is that there's actually a lot of misinformation, active misinformation about human ancestry, these misconceptions about differences between human groups, which have been around for a long time. They're very hard enduring, and notions of scientific racism haven't gone away. In fact, in some ways they've gotten. The internet and social media have helped to amplify these kinds of ideas. So we're really sort of fighting against that in some ways, when you're trying to talk about what our understanding of human ancestry, and also how, what the differences between humans mean and what their consequences are and are not. And so I think that actually is one of the biggest challenges in my field. Is trying to sort of you really what you'd like to be doing is just telling the positive and exciting story of the things that we've discovered, and yet you find that you're having to sort of overcome at the same time this sort of noisy chorus of misrepresentation, which often has quite kind of deep roots and has affected a lot of people's understanding of the differences between humans. So those are challenges, anyway, that we face.
Yeah. I think paleontology is a lot easier.
Yeah, glad.
I mean, it doesn't matter how complicated your analyses are, you can tie it back to the fossils.
Yeah.
And I mean, it's a lot harder for early animals than it is for people who study dinosaurs. But they're still relatively charismatic. You can show people different fossils. You can tie it back into very first animals. And so you do tend to get, for example, a lot of press coverage on paleontology. You have kind of BBC shows that it is out there. And I think for me as as an ediacaran paleontologist, the trick is kind of helping people to understand that actually animals did originate before the Cambrian, because people will often just talk about the first animals of the Cambrian. It's like, well, actually, things don't pop into being, incredibly complex with lots of legs and lots of eyes. It starts before then. So it's slowly and surely through events like this, through the media, both social media and traditional media, and also through museums. So we do really, really well with museums. The museums in Cambridge are fantastic. I've got the Sedgwick Museum and the University Museum of Zoology, and it's a wonderful way to kind of link in a kind of fossil past, our histories, to what we see today. So I think, relatively easy compared to these folks. Yeah.
I think within the field of plant science, crop science, one of the big kind of communication disasters was the sort of GMO controversy in the sort of 90s, 2000s, but still is kind of rumbling on. And in that case, it was, there was a lot of misconception about genetics, what these plants that were being produced were, what the risks were or weren't. But ultimately, it's held back research, and it's held back what could actually be very sustainable technology that's much better for the environment. And so engaging, about ideas of genetics, I think, is very important. But things are moving on, and the last government introduced something called the Precision Breeding Act, which is allowing more genetic modification of crops to happen. And also, things are moving even faster towards things like synthetic chromosomes. That's where, people are now able to build up chromosomes from scratch and specify, the complete sequence. So, you think about the possibilities of that technology. It's a sort of very extreme form of GMO. But without, public engagement and understanding, everyone has to come along for the ride and agree that those technologies are worthwhile or not. So, engaging with people is, I think, important for that reason.
Thank you. Do we have any questions from the audience? Thank you.
Yes, thank you. It's a question for the first speaker, really. Well, first one's an observation. Aren't you just pushing Darwin's problem back an era by going back from the Cambrian explosion to finding sort of creatures they came from, which still seem to be very diverse in the late Precambrian? And the second question is, it seems to me I can understand how we can possibly have lost the fractal kind of animals, but why do you think we lost the 3- and 4-fold symmetry, but we kept the 5 and the 2? Any ideas?
So I think for the first question, when we think things like trilobites, those are hugely, hugely complex. But when you go back in the Ediacaran, the very first Ediacaran organisms we see are relatively simple in terms of their body plans. But you see kind of simpler versions of things like trilobites slowly kind of at various points throughout the Ediacaran. So it's not the trilobites, there's a kind of upview for trilobites and other organisms. So you see it with sponges, you see it with cnidarians, you see a kind of a whole host of the major groups. You see essentially the stem groups, the very early versions in the Ediacarans in various shapes and forms. So it is more of a gentle kind of going into it. In terms of why we've lost so many of our different radial symmetries, I have no idea. I wish I did. It's a fascinating question to me because it doesn't feel like there's a good reason for it, but yeah, we have.
Thanks for really interesting talks. When you research your family tree, you often find yourself sort of going back to somewhere like agricultural labourers in one area of the world, and then you just get stuck there. And so it looks like there's very low amounts of movement. So looking at your research, how much can we infer about the actual movement of people that are causing some of these sort of common genetic traits? And is it the case that you need maybe just a very small percentage of the population to move for the traits to then kind of go into other areas, or can we infer that there's actually lots more population movement than we might believe was happening hundreds of years ago?
So the answer is sort of yes to both of those, in the sense that yes, you do. It turns out you do. Even a small amount of movement or gene flow from one region to another will actually mix ancestry quite successfully over many generations. And you have to remember we're usually looking at quite long periods of time, sort of tens or hundreds of generations in a given area when you think about what's happening there. So yes. A lot of these movements that I'm talking about could be due to some relatively small and numbers of people involved. However, there are occasions where we clearly have had an enormous change as a result in ancestry, and that could, of course, have been due to quite dramatic events like a large arrival of a number of people over the course of a generation or two, or it could have been something which took a long time. But the net result in some parts of the world is that ancestry has almost been completely replaced. And this is one of them, actually. People living here have very little ancestry going back beyond, say, about 5 to, well, maybe 10,000 years ago. Prior to that, you really see quite different ancestry of the samples that we find. And that's not uncommon. There's other parts of the world where we see that too, due to the movements of people that have occurred. So it could be that just a lot of small factors, but it could also be that every now and then there are substantial movements. I mean, we know that some did indeed happen, certainly in historical times, so they probably happened previously as well. Actually being able to detect those is one of the things that we're just being able to start, we're developing methods to do now, so it's a really exciting question.
Gentlemen.
So if life were to be found on another planet, how common do you think these processes are? How much similarity we would be expecting? What's in common or what's not in common? Assuming similar, even if there was a different evolutionary phase that you encountered that life, what would we see?
You're probably the best person to do this.
So I'll take this question. So there's a Levi-Hume Centre for Life in the Universe here in Cambridge, of which I'm co-director of, and we're a group of people kind of across all the sciences and also theology and philosophy that are trying to think about life in the universe in terms of detecting it, understanding where it comes from, where it's going. And I think what's really, really exciting from an evolutionary biology perspective is right now we've only got Earth. We're at an n=1 problem. But if we can start detecting life on other planets, then we can start getting glimpses of evolution, how it works on different planets, sometimes hundreds of light years away from us. So the James Webb Telescope is doing a fantastic job, so looking at atmospheric signatures. And so last September, a colleague of ours, Professor Madhusan, managed to resolve, for the first time on a habitable planet, K218b, carbon molecules, so carbon dioxide and methane, and hints of other molecules which are only produced by life. And so it's going to get more data and see what we find. But the fact that we've got this capability to potentially detect in atmospheres signatures of life means that we can start thinking about how kind of unusual or special or not evolution here on Earth is. So I think we're at a really, really exciting time when it comes to thinking about life outside Earth.
It probably would have a hereditary molecule there, some sort of. Yeah. It might not be DNA, but it would be something similar.
Supplementary on that, do we see the same paths evolving differently in different parts of the world? So similar theory, were different solutions found, or what you're saying right back then was one?
It appears there's one origin of extant things on Earth, because things like the genetic code are identical. There are various fairly deep similarities. It doesn't mean there weren't other alternative forms early on that perished, you know.
But it's also true that lots of things have evolved separately in different parts of the world, solutions to the same problem, sometimes the same solution to the same problem. So there are things like that evolution has found multiple similar solutions to things, and so presumably this kind of phenomena would also be the case elsewhere in the universe. It'd be very exciting to see.
I mean, I think for me, the really big question is whether life elsewhere is just microbial. So most of life on Earth now, most of the history of life on Earth has just been microbial. And is that just kind of normal? Or, you know, are we going to get animals? Often when we think of, you know, complex life, we think of plants, we think of animals or fungi, but that's not the majority, you know, either historically or kind of, I guess, in terms of biomass now. And so that's what's a really interesting question that potentially we might be able to get insights in over the next decade or two. But yeah.
How possible do you think it would be for people to actually use genetic intervention to change the race in order to allow them to go to other planets or to improve the people on this earth? I mean, I think some work has been done on trying to eliminate genetic diseases, hasn't there? I mean, it's a question of should as well as could to intervene in improving the species.
Well, it's very difficult to be clear about what we mean by improving to start with. So it's not even that itself is not a straightforward thing. But definitely, there's a lot of research in trying to cure genetic diseases, which are generally most of which are rare conditions. But there are genetic factors which contribute to most diseases, and some of those are common conditions. And so that's basically the nature, you know, a huge amount of medical research is devoted to that right now. Whether or not you would change, so to the extent to which that involves actually changing the genome, to some extent everything we do has that effect because we're still evolving in many ways, and therefore our genomes will continue to change as we go, and it will be due to the things that we do, the environment that we create for ourselves and adjust. Actually editing the genome is something which is quite, that's not something that we are clear about how to do at all, despite the claims of some people. I mean, we can do it physically, but knowing what the changes that you might make, the edits that you might make to a genome, and what the consequences would be is potentially very, very complicated. And therefore, we're quite some way away from being able to be confident, except in some very rare cases. There are some very specific diseases where we're very confident that it's just one thing, and if we make this change early on, then it will have, it will essentially cure this genetic disease. But that's a very small minority of the cases. just doing things like improving our ability to do various things or to breed on other plants or something like that would be something that that's for now completely science fiction.
Yes, because presumably genes have changed in the past as a result of evolution, haven't they? But that's
Indeed.
quite a slow progress, whereas you can speed it up by intervention.
Yes, but the problem is that the network of interactions between genes is so complicated, and a single genetic change could have multiple effects, not just on the thing that we've discovered, that we've associated with, which we don't know about. That at the moment, we really you'd be very reluctant, I think, to do it, and we should be very reluctant to do that experiment.
Someone like Hitler might not have been.
Well, yes, but yeah, not a good example.
I mean, if I could just add, we think of evolution as being very, very slow, but it can happen very, very quickly. There are some very famous examples. For example, anno lizards in the Caribbean. We have the same species on different islands, and they all adapt to the same environment in the same way. So you get different species evolving the same, kind of leg lengths, for example, and colours, depending on where they're living in the tree. So even though they're totally different species, over the course of relatively short time, so 15, 20 years, you see them evolving and changing. So yeah, most of the time it's quite short. But if you have enough pressure, evolutionary pressures, and you can get it happening relatively fast.
I think we have two final questions, so we'll just grab the microphone. Thank you. There's a lady up here, yes, and then this gentleman. Thank you.
Thank you. It's a bit late to ask about this, but what about inheritance of acquired characteristics and non-genomic influences, mitochondrial, etc.?
There is quite well documented in plants, so the environment having effects that are heritable. It seems to be less the case in humans, as far as I know. Mm-hmm. It doesn't, sort of what you might call Lamarckian type changes, don't really seem to be very prevalent in humans. But in plants, it can happen. They're quite responsive to the environment, and in some cases can kind of carry a memory of it. It is to do with the genome. It's sometimes called epigenetics, so addition of things like methyl marks to the DNA or the proteins with the DNA. But it gets a lot of press, but certainly in humans, it doesn't appear to be very common.
I'd say, in fact, I don't think there's any evidence at all, really, in human of it being a process in humans. Certainly not one that has any kind of evolutionary significance.
And the final question. Thank you.
Thank you. I think I'd like to try and play off Professor Henderson and Dr. Scally. So, Professor Henderson, you talked about GMOs, and my sense of that is that you may be sort of supportive of genetic manipulation there. Whereas, in fact, we just heard from Dr. Scally about the fact that you might have, you make one change, and the way that interacts with a network of genes might have rather unanticipatable consequences. So, maybe how do we resolve that question? Or are there ways that, for example, in plant genetics, that you can be relatively certain about the kind of effects that these changes might make?
Yeah, well, I'll just jump in. In essence, the reason is that you can do experiments with plants to see the consequences that you cannot possibly do in humans. It'd be completely unethical. And that's really the number one issue. And so with humans, even your first experiment, you're dealing with a human life, the consequences of which could be something terrible. Whereas in plants, we, at the moment, don't hold an Arabidopsis plant or anything in quite the same way. I mean, so that would be one major reason why it's a different prospect. But maybe you have another take.
I do actually hold an Arabidopsis plant.
Yeah,
in that way.
just as important.
But actually, well, if you think back to, say, the domestication of wheat, this happened in the Middle East, maybe tens of thousands of years ago. That involved, multiple species hybridizations, lots of kind of so-called unconscious selection or kind of early steps of domestication. Massive genetic change. No one filled out a health and safety form on that. And actually, with some of these techniques like CRISPR, you're talking about much more precise, pinpoint changes. And I think what Aylwyn said is true as well. Like, it's more tractable. You can really test things out to see whether there's been any inadvertent negative effects. Of course, you don't really know until you put something in the wild. You don't really know what's going to happen, and ecology is obviously very complex. So, but I think with the changing climate, sustainability, genetic modification can help you kind of meet those challenges, and I think it's something we should be using more in a careful way. But currently, partly because of the GMO controversy, a lot of people's hands are tied essentially in terms of what you can do.
I think we will close it up there. So obviously we've got 3 exceptional speakers here and researchers, and we've only really scratched the surface. So make sure you go to the Museum of Zoology. Make sure that you tell everyone that you're related to more than just a king or more than just 1% Viking. I like to know that I'm related to Cleopatra. That sounds lovely. And also make sure that you read more and change your ideas about genetics and what that means for the universe and for us moving forward. Please join me in thanking our speakers. Thank you so much.